All right, let's dive into a crucial process happening inside your cells right this very second.
Just imagine, what if your body's cellular power source just started to decay? It would be a total disaster.
But luckily, evolution came up with a pretty clever way to stop that from happening.
It all has to do with protecting our mitochondrial DNA, or mDNA, the tiny genome that fuels our cells.
Here's the kicker. You don't get this from both parents. You inherit mtDNA only from your mother.
And inheriting it this way is risky. It means harmful mutations could just pile up over time.
So how do our cells possibly prevent this genetic train wreck? That's been a huge mystery.
Well, for years, we've known about two main defenses, something called a genetic bottleneck and a filtering process.
First up, the bottleneck. It works by drastically cutting down the number of mDNA copies that get passed on.
So, a mother's cells have a mix of mtDNA. Some of it's healthy, some of it's mutated. But because of the bottleneck, only a small, random sample of that actually makes it to her child.
At the same time, this purifying selection acts like a quality control inspector, actively tossing out damaged copies.
So we've got these two systems. The big question was, how did they actually work together?
And now, a huge breakthrough from the Karolinska Institute has finally connected the dots.
Turns out, a smaller bottleneck actually makes that quality control process way more effective.
As the lead researcher put it, the size of that bottleneck is the key factor for filtering out mutations.
And this whole filtering process is tied to something called autophagy. That's the cell's own recycling program.
Okay, so this is all pretty cool science, but why is it a big deal for you?
Because when your mt.tn quality goes down, it's linked to a whole host of serious problems.
Understanding this link between the bottleneck and quality control, well, it opens up brand new doors for research.
And that, of course, could point the way to future treatments for these mitochondrial diseases. Which leaves us with a pretty fascinating thought. Could we one day tighten this bottleneck to stop diseases